So thank you. We had a short report in Hematologica describing both spatial and temporal heterogeneity in diffuse large B-cell lymphoma, I think, which raises some really interesting themes. And this was a young woman who presented with stage 3 disease, and biopsy of the disease below the diaphragm had revealed a germinal center phenotype. However, after having had R-CHOP chemotherapy, the disease in the inguinal region regressed very nicely. However, the disease that she had above the diaphragm unfortunately progressed. And when this was re-evaluated, this was a diffuse large B-cell lymphoma of the ABC phenotype. Now, we looked at exomic sequencing in these for the pattern of mutations, and we saw that the disease at presentation and the disease at progression had some patterns of mutations that were shared, suggesting, obviously indicating a similar origin. But we recognized that there was divergence in the clone, and the disease at presentation had genes that were mutated in epigenetic regulators. However, the clone at progression demonstrated genes that were dysregulated in the NF-kappa B pathway, so it acquired mutations along that pathway and had acquired a P53 mutation. So I think what's really interesting is to show that there is diversification of the clones, and there is intratumoral heterogeneity. So I think this is important in terms of our clinical need to biopsy patients and to understand that necessarily what you may biopsy may not be representative of the whole tumor. So that leads me on really very nicely to talking about changes in our understanding of the classification of diffuse large B-cell lymphoma based upon molecular data. Last year, we were able to publish data on a subset of GCB-type diffuse large B-cell lymphoma that had particularly poor risk features and did poorly. And we called this a molecular high-grade signature, MHG. And this identified a group of patients that included those who had dual translocations in MYC and BCL2, but also expanded the size of that class or that category to patients who didn't have the translocations evident by FISH analysis, but behaved as if they did have so. So we've identified a risk of a group of patients with high-risk disease that do much worse than the standard GCB, and interestingly, may be appropriate for specific therapeutic targeting, because we saw that there was an effect of the addition of bortezomib to R-CHOP chemotherapy in this group when we analyzed the data retrospectively from the Remodel B dataset. At the same time, the group from British Columbia described a double hit lymphoma signature where they had derived a set of genes from that defined a group of patients that did contain a double hit by FISH analysis, and that when that signature was applied to a broader group of GCB type diffuse large B-cell lymphoma, then it identified a larger number than those who just had the double hit, exactly as we had done in the MHG. And there were some clear similarities between our group of patients and the group of patients that have been identified by the double hit signature. So once again, this is refinement of the classification and us being able to better understand which patients are at particular high risk and may potentially be amenable to more targeted therapies. So what's very clear from the data that has come out regarding diffuse large B-cell lymphoma is that the simple transcriptome-based classification of diffuse large B-cell lymphoma according to the cell of origin model, i.e. the 3 classifications of germinal center-like, GCB-like, activated B-cell-like, and those unclassifiables, really don't account for the heterogeneity that we see amongst the patients, both molecularly and also in terms of clinical behavior when treated with R-CHOP chemotherapy. And over the last year or a little bit more, we have seen reclassification and the establishment of at least 7 clear subclasses of diffuse large B-cell lymphoma based upon integration of data from multi-omic platforms to establish different classifiers. And we've seen recent data from George Wright at the NCI, who works in Lou Stout's group and published in Cell, Cancer Cell, that we see a group of, we see clear classification of patients with diffuse large B-cell lymphoma, and that we're able to clearly probabilistically classify them into different groups. And we see that there's refinement of the ABC subgroup with degrees of risk depending on the molecular profile, refinement of the GCP group, again with different risk, and that becomes clinically relevant. And because the pattern of oncogenic aberrations in these different 7 categories may be amenable to therapeutic targeting. So I think these are really exciting times. However, I think what's really clear for us working in the clinical trials community is that we need to be able to classify these patients in a clinically meaningful time frame. So what we do need to do is we need to be able to integrate the data from these various platforms that we're analyzing to be able to ascribe a patient to a particular molecular subclassification and have the result available within a time frame so that we are able to perhaps target the phenotype genotype of that disorder. So I think this is really exciting times for us as we be able to unravel and unknit that heterogeneity, but what we need to do is make sure that we can do that in a clinically relevant time frame.